You know, every single living thing, from a tiny bacterium all the way to us, runs on one master instruction manual, our DNA. But here's the thing, copying that manual, which in humans is over 3 billion letters long, is one of the trickiest challenges a cell ever has to face. It's a process just filled with these incredible puzzles, and as we'll see, life has come up with some truly stunning solutions.
So, how in the world does a cell pull this off? How does it make sure the copy is perfect, that it happens at exactly the right time, and that it doesn't accidentally shred the original instructions in the process? We're going to dive into 3 of these big fundamental puzzles and explore the absolutely ingenious molecular machines that life evolved to solve them.
Okay, so our first puzzle is all about timing. I want you to imagine you have to copy a gigantic book. But here's the rule. You have to make exactly 1 copy, not 0, and definitely not 2. For a cell, getting this wrong, even by a little bit, is a total disaster. This is what's known as the only once problem. So a cell needs a completely foolproof system, right? A system to say, go, start copying, and then immediately lock the door behind it so it can't start again until the next cycle. How does it flip that switch from wait to go, and then back to wait again? Well, it turns out the secret is written right onto the DNA itself.
This number right here, 11, this is the key. In the bacterium E. coli, there's a specific starting line for replication called ORAC. And what's wild is that this tiny little region is packed with a specific DNA sequence, GATC. It's got 11 of them, which is way more than you'd ever find by pure chance. This is no accident. These are the chemical flags that control the entire operation.
The whole solution works kind of like a single-use ticket for a concert. When those GATC sites on both strands of the DNA have a tiny chemical tag, it's called a methyl group, the ticket is valid. The go signal is on, and replication machinery can get to work. But as soon as that section is copied, only the original parent strand still has the tag. The brand new strand is bare. This half-tagged state, or hemimethylated state, is like a used ticket stub. The signal is now off. No re-entry. So let's walk through it. Step 1. The go signal is on, replication kicks off. Step 2. The moment that new half-methylated DNA is made, it's immediately grabbed and physically hidden away at the cell membrane. Now, this is absolutely crucial. It stops the machinery from jumping back on and starting another copy. And then finally, step 3. After a built-in delay, another enzyme comes along and adds the missing tags to the new strand. The ticket is reset, fresh and ready for the next time the cell needs to divide. It's just a simple, elegant, self-regulating clock.
All right, so that elegant clockwork is how cells play by the rules. But our next puzzle is all about what happens when you break them. For decades, the central dogma of biology was seen as this unbreakable law. Information flows one way, from DNA to RNA to protein. But it turns out some of the most cunning invaders in the biological world, I'm talking about retroviruses, found a way to run the whole process backwards. Back in 1970, 2 scientists, Howard Temin and David Baltimore, they independently discovered an enzyme that provided, and this is their term, dogma-shaking proof that genetic info could flow backwards. They found a biological machine that could do what was thought to be impossible. It could read an RNA blueprint and write it back into DNA. They called this amazing enzyme reverse transcriptase.
So here's the hijack plan. A retrovirus, like HIV for example, injects its RNA and its special reverse transcriptase enzyme into a host cell. That enzyme immediately gets to work, building a DNA copy of the virus's RNA genes. And then, this new piece of viral DNA gets stitched directly into the host cell's own chromosome. It becomes a permanent, hidden part of the cell's blueprint, just waiting to make more viruses.
But here's the real twist. Reverse transcriptase is fast, but man, is it sloppy. It makes a mistake about once every 20,000 letters it copies. That error rate is massive compared to our own hyper-accurate DNA copying enzymes. But this sloppiness, this is actually the virus's secret weapon. It means the virus is constantly changing, constantly mutating, which is what allows it to evolve so rapidly and stay one step ahead of our immune systems.
Okay, for our third and final puzzle, we're coming back home to our own cells and a really tricky problem with our linear chromosomes. You see, every time a cell divides, the copying machinery runs into a mechanical issue. It just can't quite copy the very, very end of the DNA strand. This sets up a kind of cellular countdown timer.
This is what we call the end replication problem. You can think of it like a shoelace that gets just a little bit more frayed each time you tie it. With every single cell division, the chromosome gets a tiny bit shorter, and over time, that could be a huge problem because it could start eating away at important, essential genes. So, how does life deal with this?
Well, the first part of the solution is just brilliant in its simplicity. You just put something disposable at the ends. These disposable ends are called telomeres. In humans, it's the sequence TTAGG, just repeated thousands of times. This creates this protective buffer of junk DNA, basically. The chromosome can afford to lose a bit of this cap without doing any damage to the important genetic code that's stored inside.
But just having a disposable cap isn't going to cut it for cells that need to divide over and over again, like our reproductive cells. They need a way to actually fight back against this shortening process. And this is where the hero of our story comes in, an incredible enzyme called telomerase, which was discovered by Elizabeth Blackburn and Carol Greider. It's a machine that can actually rebuild the ends of our chromosomes.
So how does it pull this off? This is the real magic trick. Telomerase actually carries its own instruction manual, a little piece of RNA that it uses as a template. It latches on to the very end of the chromosome, and using its template, it just adds on more and more of those TTAGG repeats, extending the strand. Then the standard replication machinery comes in and fills in the other side. It actively adds length back, completely defying that fraying process.
Now, this whole puzzle, it's not just some obscure molecular curiosity. The solution, this enzyme, telomerase, is directly connected to some of the most profound parts of our own lives: why we age, how cancer is able to grow, and the very idea of cellular immortality.
And this right here really shows you the two-sided coin of telomerase. In most of our regular body cells, the gene for telomerase is switched off. So as our cells divide and divide, our telomeres get shorter and shorter. And that's a key reason we age. But in our germline cells, the ones that make the next generation, and pretty ominously, in about 90% of all cancers, telomerase is switched on. This is what allows them to blow right past the normal aging clock and divide forever.
And this isn't just a theory. It's incredibly tangible. I mean, look at these numbers. They show the average length of a person's telomeres shrinking from around 11,000 base pairs at birth all the way down to less than 4,000 in old age. You're literally watching the cellular clock tick down over a lifetime. Eventually, this shortening triggers what's called senescence, the point where most of our cells get the signal to just stop dividing for good.
So we've seen these incredible solutions to life's biggest replication puzzles: a bacterial timer, a viral rule breaker, and a machine that literally battles aging at the tips of our chromosomes. And it all leads to one final, profound question. We've basically found the switch for cellular immortality. What happens when we finally learn how to control it? And maybe more importantly, should we?